Hello and welcome to the International Postdoc Forum for the Philosophy of Science, hosted by the Minnesota Center for Philosophy of Science. My name is Alan Love. I'm the director of the Minnesota Center for Philosophy of Science, and it's my great pleasure to be with you today. Just a couple words about the Postdoc Forum. It was something that began during the pandemic when we were all online, and we were interested in giving opportunities to recent PhDs and emerging scholars, opportunities to present their work where they had potentially lost that due to conference and workshop cancellations and restrictions on travel. And so we introduced the program at that time, but the response was very positive, and we thought there was good reason to continue it because it was really a good thing. So we're showcasing virtual research presentations from international scholars with commentaries from local community members here at the University of Minnesota and Minnesota Center for Philosophy of Science. So I'm very delighted today to introduce Yasmin Haddad, who is an FRQSC Postdoctoral Fellow from the University of Quebec in Montreal. The title of her talk is Measuring Ancestry, and her commentator, Maria Nieves Colon, is from the Department of Anthropology in the University of Minnesota. I will introduce her briefly when the commentary comes up after the talk. But now I'm going to turn things over to Yasmin to tell us about measuring ancestry. The floor is yours, Yasmin.
Okay, so I'm going to start sharing my screen. Just 1 second. Okay, so for some reason, 1 second. When I share my screen, I'm seeing everything black when I put it in full screen mode. I don't know why.
Yeah, that we're seeing the black also. So
okay, I don't know. It worked fine earlier. Maybe just give me 1 second and I'll just download it as a PDF. Sorry about that. The test worked fine.
No problem.
Let's try again.
Yep, that's coming through fine. We can see it.
Okay, now I just want to try and make it like, full screen. But I guess.
Yeah, I think that would be under view for view full screen.
Is that okay?
Oh, there it is. Yep.
Great.
Perfect.
Okay. Sorry about that. Now let's see. Okay, that works. Okay. Okay, so well, thanks a lot, Alan, for organizing the event, and thank you, Maria, for agreeing to be a commentator. I really look forward to the discussion and to the feedback I might get on this work. So I'll just start by briefly outlining the structure of my presentation. First, I'm just going to briefly introduce the notions of ancestry and the context from which I'm writing this paper at the moment. I'll go over some of the goals that I have with this specific project. And then I'm going to talk a little bit about why I think it's interesting and fruitful to think about ancestry from a measurement perspective. And then I'm going to talk about some ways of measuring ancestry. And the core of my talk will be some of the challenges that I highlighted from a philosophy of measurement perspective that a lot of scientists, geneticists are encountering when measuring ancestry. And then I will maybe give some ideas as to the outlook. So, some suggestions on how we can think about these challenges and eventually move on from them. My goal is not to provide all the solutions, but really just to show how, you know, philosophers of science can take interest in this question and contribute to the current debates that are going on. And then I'll finish with some very brief concluding remarks. So I just want to mention that this is very much work in progress. So I've just started working on this project as part of a project that's funded by the FRQSC, which is the Quebec Research Fund. And this is it's going to be more or less a 3-year project. So this paper is really the first step in the construction of this idea, alongside a couple of other papers I've been working on with a collaborator. And I'm really looking forward to putting these ideas out there in the world and, you know, seeing what happens with them. And I think this talk is just a great forum and a great opportunity to do so. Okay, so first of all, I want to just talk a little bit about some of the terminology I'm going to be using, because I imagine not everyone is familiar with the idea of ancestry, but I'm interested in ancestry in the context of human population genomics. And broadly, very broadly, we can define genetic ancestry as the path through an individual's family tree by which they have inherited DNA from specific ancestors. Now, genetic ancestry is sometimes classified or organized in terms of groups, which are called genetic ancestry groups. And these groups are groups of individuals that are considered to be genetically similar in some respect. So depending on the study, what the similarities will vary. And they're most often grouped in terms of geographic or continental origin. So the issue here is that when we're talking about genetic ancestry groups, the most common way of grouping individuals that are genetically similar is through using some labels that refer to a continental origin or geographic origin. For example, we can say European ancestry or East Asian, or there are many different ways of classifying genetic ancestry groups, and it will vary depending on the study. Now, what's interesting about this is that really measures of ancestry are kind of a proxy for genetic similarity. And genetic similarity is important because it can be informative of risk factors, for example. So again, it depends on the type of study. I'm going to focus more on studies that are interested in either gene discovery for the origin of complex diseases or just studies that at the end want to try to establish some kind of information that will be useful for, targeted medicine or doctors or pharmacogenomics. And anyway, so something that has a clear application. Now, there are many other subfields in genomics that are a lot more exploratory, that are just kind of trying to discover genes for X, for example, but here I think that ultimately I'm interested in, what is this information going to be used for? So why talk about ancestry now? And really, I've seen in the past few years, let's say in the past 2 years, 3 years, a lot of interest in the idea in the concept of ancestry, which in and of itself appears differently in different studies. So people define it slightly differently, but what's common to the interest in ancestry is that a lot of scientists and social scientists are really curious and investigating, if ancestry is being used very differently from other notions that appear in genomics, such as ethnicity and race, for example. And we all know that race in genomics is quite a loaded and problematic term, and ancestry has kind of emerged as an alternative that is more like related to a statistical label rather than a social label. And this is interesting because sometimes ancestry is presented as almost like neutral, but we know that there is a lot of slippage between concepts like ancestry and race. And it's interesting to kind of talk about ancestry in this dimension and to evaluate the slippage, whether it's warranted or not, and to see how ancestry is being used in those studies. Now, another reason for talking about ancestry is that recently the National Academy of Sciences has kind of organized a committee to discuss the use of population descriptors in genomics. So Ancestry is a population descriptor. That means we can describe populations in terms of genetic ancestry. Race is another example of a population descriptor. Ethnicity is another example of a population descriptor. And the goal of this committee was to think of the best practices to describe populations in genomics, which descriptors should be used and which shouldn't be used. So in the recommended approaches for the use of population descriptors, this committee kind of produced this table, which I find really interesting because you can see here the red kind of indication says that this descriptor should not be used, and here we have the type of study that we're talking about on the left column. So for most genomic studies, they are recommending not to use race as a descriptor. And then if you look at genetic ancestry here, for most genomic studies, we can see a question mark. And I think that the question mark here refers in some cases, it's not clear whether it should be used, or it depends on the outcome of the study, on the goal of the study. And as philosophers, when we see a question mark, we're like, okay, there's something there that's interesting. There's something there that's worthy of thinking about, worthy of investigation. So really my idea with this project came when I saw basically, let's say 4 or 5 question marks in a row. I was like, okay, there's something there that could be interesting for us. And this report was published last year, so the discussion is very recent. I sat in all of the panels of all of the public sessions of the discussion and the deliberation, and really it was such a great opportunity to have this accessible, because that was a very rich environment in which to start thinking about and learning more about how this descriptor is used. Now, Ancestry, as I said, has increasingly replaced the descriptor race over the past decades. Whether the substitution is warranted remains a topic of discussion, and I would say not only discussion, but even controversy. So in the literature, you can find those that will really defend the usefulness of Ancestry, and on one spectrum, and then on the other end of the spectrum, you have calls for just not using Ancestry and just talking about similarity or relatedness. And I'll go back to this point a little bit later in the talk. Among the many challenges with choosing descriptors, there are challenges related to measurement, and those specific challenges remain unexplored. And this is what I want to tackle. This is the angle I'm choosing to tackle the question of ancestry in this particular presentation today and this project. So what are my aims and goals? First, I want to untangle one aspect of using ancestry, because there are many aspects. So we could talk about the political aspects, the social aspects, but here I want to talk about ancestry as a descriptor and how it's being measured. So how are we really assessing what are good measures of ancestry? Second, I want to apply insights from the philosophy of science, more specifically the philosophy of measurement, the tools that philosophers of measurement have come up with to investigate and clarify the challenges to measuring ancestry. And finally, I want to think about how to mitigate those challenges at different scales, such as the individual, the social, and the political level. And I'll go back, this will be more towards the end of the talk, but really the core will be aims 1 and 2. And at the end, I will talk about a little bit about how we can mitigate those challenges. So why measurement? Why do I think it's a good idea to talk about measurement? Well, if we take a very general definition from the Stanford Encyclopedia of Philosophy, we can see that most, but not all, contemporary authors agree that measurement is an activity that involves interaction with a concrete system with the aim of representing aspects of that system in abstract terms. For example, in terms of classes, numbers, vectors, and so on. So this is just a very, very general definition for us to get started with the question of measurement. Now, when we are talking about ancestry, we have this particular setup where we have scientists interacting with a concrete system, and the goal is to represent some kind of relationship between parts of that system. Scientists are within a community. They are constrained by funding agencies, by census requirements, by external constraints such as political context, for example. When they're interacting with a concrete system, Mostly when we're talking about ancestry, then the kinds of study that I'm focusing on, we are talking an individual's DNA. More specifically, we're talking single nucleotide polymorphisms, which are one way of assessing variation between individuals. That is quite informative. And here they're representing this information that's being measured through different metrics, but most often we see degrees of genetic similarity that is frequently, and this degree of genetic similarity is frequently expressed in terms of shared genetic ancestry. So the idea here is two individuals that are genetically similar, they will also share ancestry and they will tend to be grouped together in a cluster or a group or a population. So better measurement, we know, is tied to empirical progress in any scientific discipline. So as metrology evolves and we have better ways of measuring what we're trying to study, then we know that it's tied to empirical progress. But interestingly, as Hule and Al have mentioned in a really, really rich discussion, there's kind of a lack of a theory of measurement and meaning in biology. I think this is changing. I think it really is changing. But when we compare, for example, the literature on measurement in philosophy of biology and compared to other science like physics or psychometrics or even psychiatry, I guess the literature and the work in these other fields is a lot more, there's a lot more to find on those fields than on biology itself. So I think it's interesting to tackle this at least from one in one discipline in the biological sciences, which in my case is genomics. And Hule also say that, you know, the representational measurement theory is a useful tool for biology because RMT, as it's called, is assessing whether relationships among numerical measurements are consistent with the relationships they are meant to represent. So it's the idea, am I measuring correctly what I wish to represent? And this is really a core question for the examples that I'm going to be talking about later on. So what's the relationship, and you know, what do we mean when we say measure ancestry? What do I mean when I imagine measuring ancestry, and what do I see in the literature? Well, there's a huge overlap between shared ancestry and genetic similarity, because shared ancestry is a measure of genetic similarity and relatedness. And this is the point made by Graham Coop in his 2023 paper, where he really says, you know, we should just move away from talking about genetic ancestry, and instead we should just talk about similarity and relatedness. A sampled individual is associated to a source population based on this relatedness of similarity, and they're grouped into different populations depending on how many individuals are sampled, and the populations are themselves modeling constructs and simplifications that are based on a lot of background assumptions. So what counts as a population here is crucial. And I mean, there's a lot of work in philosophy of biology about that specifically. I want to especially to talk about the work of, just briefly mention the work of Lisa Gannett, where she has a paper which really talks about this idea of population thinking and how it's related to typological thinking. I mean, especially with the concept of population, philosophy of biology is really rich of examples and contributions about that. So what are some of the challenges that I have found while studying this topic? Well, basically, there are three main challenges to measuring ancestry, and this is what I would like to delve into now. The first is a problem that's well known for philosophers of science, which is the problem of coordination. The second is a problem that's very well known to philosophers of measurement, which is the problem of bias. And the third is a problem that's very well known for both philosophers of measurement and philosophers of interested in data, which is the question of legacy data. So I'll approach these three in this order, starting by the problem of coordination. So very broadly, The problem of coordination is a classic problem in the philosophy of science and the philosophy of measurement, where what is measured is usually coordinated through a theoretical concept. So we know that, for example, when we're trying to measure something, the techniques and the instruments that we're going to use to measure this thing are going to be shaped by previous theoretical commitments. And at the same time, the only way to test the theory is to measure using those same instruments. So that creates a kind of circularity. I would say almost like a vicious circularity, because it's impossible to untangle measurement from theory. And the correct term would be that measurement is theory laden, because it always depends on a lot of background assumptions. Now, this circularity in and of itself is not necessarily a problem. In fact, one may say, well, this is only a problem for philosophers of science, because in practice, scientists are pretty much okay with the idea of that this measurement is circular with theory, and that this doesn't pose a major problem. But still, I think that it's interesting to address the problem of coordination in this case, because there is this kind of circularity between measuring similarity and the assumptions that we have about ancestry. So, for example, the theory that we have, the assumptions that we have when we are trying to establish what a population is, and the assumptions that we have about human migration histories and movements between continents and so on. So all of these will shape, and also assumptions about geography, because really we are using a lot continental regions. So the idea here is to understand, what comes first, but not only what comes first, but like how should theory influence measurement, and in which cases it probably should be neutralized, or at least we should try to think about it outside of this loop. So here we have a formulation by Baswan-Prasen of the problem of coordination, where the rules or principles of coordination that can be introduced to define particular sorts of measurement cannot even be formulated except in a context where some forms of measurement are already accepted and in place. So let's look at a first example of where this problem of coordination seems to be very obviously occurring, okay? And it's got to do with clustering. So imagine that in many studies that are going to collect large samples of genetic data and try to classify them and cluster individuals according to genetic similarity, the idea is to use algorithms that are called clustering algorithms. One such algorithm is called structure, and it was developed by Pritchard and al. in the year 2000, and it is really widely used, and it kind of became, as Deborah Bolnick mentions and John November mentions, it kind of became somehow a victim of its own success, because this algorithm has some limitations, but it's very, very popular, and it's used a lot to precisely cluster according to similarity. It uses a Bayesian approach that assigns individuals to source populations, and the source population is called, let's call it K. And one of the persistent challenges with that is inferring the number of source populations. So if I have a sample of, let's say, 100 individuals, what is the best way to cluster? How many populations do I want to group them into? Do I want 3 groups? Do I want 4 groups? Do I want 5 groups or 6 groups or 8 groups? In this case, another challenge is that the value of K in this specific approach with this specific algorithm has to be predetermined before. So there are some very important model choice procedures that are necessary, and usually in most publications, what happens is to try to model the data in terms of different K values. So we have, for example, K equals 4, K equals 5, K equals 6, 7, 8, and so on. In the original paper, I think if I remember correctly, they went up to K equals 18. But the problem is that increasing the value of K can lead to overfitting of the data. So there need to be other ways to verify what would be an optimal number of clusters. Here we have, I made a very, very simplified illustration. I could cluster the same population in terms of K equal 3, or I could cluster in terms of K equals 6. And it all depends on how I'm going to predefine this number of clusters. Now, this is, as I said, it's a particular case where the model works in such ways that one has to predefine the number of clusters. I'll go over another example after where we don't need to predefine the number of clusters. So here, to me, it's very obvious that we encounter a coordination challenge in this step because we have theoretical assumptions that are guiding the model choice, such as what is the optimal number of clusters and the measurement procedures themselves to assess relatedness and similarity. Additionally, we can reach an optimal number of clusters, and it will also depend on the interpretation of results for different k-values. So here, there's a very interesting paper by Bolnick in a chapter of a book actually by Deborah Bolnick in 2008, where she really goes through this particular example of clustering using the structure algorithm, and she explains why the authors decided to use the number k equals 6 and how convenient it was that k equals 6 roughly matches the number of 5 continents, and that in the end the most important reasons for using that were not necessarily because the results were better, but it was just a decision to use k equals 6. Now, of course, the paper is transparent and says, we've tried for other values of k, but it was really a model choice procedure to use k equals 6 in that particular case when there were other good results with different k values. But somebody at this point might say, well, wait a second. If you look at clustering techniques, not all clustering techniques require predetermining the number of clusters, which is true. Some studies, in fact, many studies, such as admixture studies and genome-wide association studies, which are called GWAS, will not necessarily require a predetermined number of clusters, because some studies just have an exploratory goal, right? The goal here is to say, well, I'm going to collect the data, I'm going to sample this data, and I'm going to analyze the genetic data that I have, and then I'm going to run an algorithm that's going to determine the optimal number of clusters that is representative of significant similarity. In this case, there seems to be negligible influence of theoretical and or background assumptions, because the idea is to just take the data and let an algorithm run through it. Now, there are some assumptions, but not as strong as having to predetermine the number of clusters. In this case, I'd like to talk about a very interesting study. It's an ethnographic study that was carried out by Fujimura and Rajagopalan in was published in 2011. And the authors of the study interviewed many geneticists working in genome-wide association studies that use the Eigenstrat algorithm, which is a part of the Eigensoft package. And here, one reason why they chose to use this specific algorithm is because precisely you don't have to predetermine the number of clusters. It will tell you what the optimal number is, with a degree and with options that after you can choose. But the idea here is that you don't have to have a predetermined value to run the model. How it works? Well, it generates SNP variation scores, and which are really adjusted numerical values along axes of variations that represent an individual's variation across single nucleotide polymorphisms. And the algorithms do not depend on pre-labeling or pre-sorting samples or predefining the number of clusters. So in this case, we might say, well, where's coordination here if I don't have this very strong background assumption that a certain number of clusters is important? Well, it turns out there are still some challenges, not at the stage of model choice, of trying to determine a value of k, because that stage is does not occur in this specific software. But really, the idea is that even with the results of the clustering, the software was still used to produce clusters of plots of individuals, for example, for Finland, Japan, and Sweden. And even though the plots may not be necessary, even if we could just be talking about, for example, similarity or relatedness, the samples were still organized in terms of clusters. And the interviewed researchers checked whether the plot group samples matched assumptions about the sampled individuals. So assumptions here can come in many forms. One form of those assumptions can be, for example, self-identified ethnicity at the beginning of a study. So the idea was to say, well, let's see if participants, when they sign up, they have to indicate their ethnicity. Let's say if the way we sampled and clusters matches the original ethnicities, but it also creates some kind of circularity, because if you already give a list of, let's say, 6 or 7 or 8 different ethnicities in the sign-up sheet, then and I'm sure we all have to sign these sheets somewhere, sometimes for bureaucratic purposes, the choices are already constrained. So the idea here is that these assumptions, these background assumptions, they might not come in at the stage of predetermining the number of clusters, but they do come in at a later stage when those specific researchers try to match the plot group samples with the assumptions about the sampled individuals, for example, based on self-identified ethnicity. So here I just want to say, I don't want to be naive and to say, well, let's just forget about clustering, because that's not my point here. Here as I said, I'm more interested in clarifying those challenges and putting them out there so that we can think about, how to deal with those challenges. And I'm not also trying to say that, we should just drop the general use of ancestry labels, because I think there are very practical reasons for using them. And Graham Coop summarized them very well, again, in this paper, in this 2023 paper. One major reason to keep using ancestry labels is the simplicity to share and compare results across a scientific community. Now, this is a really compelling reason, because it has influence on how the data will travel, how the data will be processed, how the community will interact. So I think the coordination challenges, they shouldn't just, I'm not just highlighting them to say, we'll forget about clustering, it's just unrealistic. But I think it's interesting to delve deeper into those techniques and try to understand what some of the challenges are. Now, the second challenge that I'm going to tackle is the question of bias. Now, I will talk about 2 types of bias. The first one is sampling bias, but this has been extensively discussed in the literature in genetics and human genomics and so on. Sampling bias occurs when members of a population are more likely to be systematically selected over others. The idea here is that also recently, the past, I would say 10 to 15 years, there's been a huge concern about lack of diversity in genomics and lack of representation of diversity in those studies. And a lot of research groups are really making it a priority to collect very diverse samples and to kind of recognize that genomics really failed on diversity, because a lot of the studies were really Eurocentric, so focusing on individuals of European ancestry. Not only that, but there's an acronym called WEIRD, like WEIRD, where most of the studies focus on individuals that are Western, educated, industrialized, and from rich developed countries, sorry. And the question here is okay, so if we are only representing a bias that is sample, if we have the sample bias in the data we're collecting, how can we make reliable predictions for people that are underrepresented? And I think this has been extensively addressed. These are just 2 examples that are recent examples of these concerns, but the literature on this is extremely rich. But I'm interested in another kind of bias that has been less addressed in this context, which is a kind of bias which is familiar in the research community about machine learning, and it's called label bias. And here, label bias occurs when in cases where the labels are inaccurate, and therefore the training data set misrepresents the target population. Here's a definition of label bias by Erantal. Label bias leads to errors that arise due to differences between the intended and the actual labels, whereby actual labels are only indirect approximations of the intended ones. So I think this is the one that I'd like to focus more with a couple of examples. Because when we talk about the choice of labels, and when we are designing any classification systems, whether it is in evolutionary biology or in the case of genomics, when we're clustering, it's a kind of classification. We will always encounter trade-offs between the stability of a classification system and the dynamic aspect of the classification system that is fit to support data journeys. So the idea here is to say, well, we need a system that has labels that are stable enough that they can be shared and that databases can be built on top of that based on those labels. But we also needed to be open to change and to self-correction and enrichment and improvement. And this is a real challenge because in a way, the more robust the classification is, the less we tend to want to tweak it or to change it. But at the same time, there is increasing recognition that those classification systems need to be periodically revised. So label bias creates another problem, which is slippage between statistical labels, which appear less arbitrary, and social labels. So this is precisely the case that happens with ancestry and race. Here we have a like ancestry is presented as a statistical label. Many scholars who defend the usefulness of ancestry say, well, this is something that we can see in the data, that pops out in the data, that we can cluster based on the data. And genetic ancestry groups represent a statistical measure, for example, European ancestry, but there is easily slippage between the idea of European ancestry and social labels such as white. Okay, so that happens with many different ancestries and many different races. And the question here is, are we using the right label given that there is slippage between statistical and social labels? So this is just a first example. A second example can be seen in this really excellent book by Catherine Nash, where she explores the idea of genetic geographies. In the first chapter and the second chapter, she goes back to this idea of using discrete labels to represent continuous variation. Now, we know that human genetic variation is continuous, and yet we tend to represent it as discrete. So we represented in terms of clusters, we represented in terms of separate populations, and most often we represent in terms of clusters that roughly map into continental regions when the data, as I explained in my first example for coordination, could fit different values of K. And in the end, what stays, especially in science communication and with the lay public, is the idea that there is some kind of discreteness to human variation that is based on continental region. This is very apparent when we're looking at results of genetic ancestry tests like 23andMe and I don't know, MyHeritage. I mean, there are many, but the idea is to really, of course, it goes into subcontinental regions in those specific cases, but it does tend to convey the idea that there's some discrete aspect to something that is in fact continuous. So we might be using discrete labels to represent something that is continuous. Finally, I want to tackle briefly the question of legacy data and what I have in mind with this. Well, legacy data is a bit of a tricky one because it's a bit like the chicken and the egg problem, who came first, you know. And genetic ancestry determination really depends on reference samples. So the idea is, let's say I'm going to try to conduct a study to learn more about some aspect of genetic variation in a diverse population. The way that one of the steps that I'll have to do is to compare my results to a reference sample. And the labeling of reference samples has implications for how the test samples are labeled. So if I want to establish a really good comparison, it's in my interest to sometimes keep using the same labels as a reference study, because then it will be able to compare the results more easily. And this is called, this is one aspect of legacy data, is existing data that is extremely useful and that has become a de facto standard. And sometimes there's a little bit of uncertainty about whether such data meets current ethical or scientific standards. So in some cases, we will still see racial labels. In some cases, we will still see some kind of arbitrary ethnic division, which is the case of, I can give you an example right now where I found the divisions to be very arbitrary. The NIH is sponsoring a really huge cohort study called All of Us, and if we look at the data quality reports, the division of the labels is quite interesting, and it's supposed to represent diversity, but I mean, I think it's important to raise questions about the choice of those labels. And there is uncertainty, as I said, about whether such data meet current ethical or scientific standards. Here's an example. Often we see the reuse of existing databases as references and little incentive to rethink classification systems that are already in place. For example, the International HapMap project is really heavily used. And in this specific case, the samples and the data are geographical labels, and researchers attribute geographic meaning to differences in SNP frequencies because this is their reference database. So as I said, often there is little incentive to rethink those systems because they're so well established, and they are considered, they become a de facto classification system. So how do we move from this? I want to give some clues. This is really the parts that I'm still working on, and that I'm looking to collaborate with scientists and people really working in the field in genetics to see what what should we be doing, how should we be thinking about those challenges. Again, I'm not trying to present the challenges so to say, okay, it's all wrong. No, absolutely not. I'm more trying to say, well, let's delve deeper into these issues and how can we think about it better and take a moment to reflect on those, and what can philosophers of science contribute to this discussion, because I often find that we are sometimes absent from this discussion, and I think it's interesting to take part in this interdisciplinary thinking process. So when we're talking about coordination, philosophers of science have a solution, especially a more pragmatic philosophers of science, such as Hafek Chang. The idea is epistemic iteration, where successive stages of knowledge build on each other to enhance the achievements of certain epistemic goals. And there are two ways to achieve epistemic iteration. The first is enrichment. That is, I'm going to refine the original system. I'm not talking about negating the whole system and starting from scratch. That's just naive and unrealistic, but rather to through time, successfully make modifications that will improve the system that I'm working with. And another one would be self-correction, which is really a strategy that we can see in the National Academies of Sciences report, because often in that report, they are talking about change and correcting and replacing racial labels for other kinds of labels such as whatever ethnicity or ancestry, but there are cases of self-correction that have already been mentioned in the literature and in these committees that are talking about the use of population descriptors such as ancestry. Self-correction requires content alteration that is a result of inquiry. So the idea here would be, well, if the results show that we shouldn't be talking about race, but we should be talking about ancestry, then we should self-correct and start using this measure instead of other population descriptors. Another point that I've been really thinking of, and I'm actually working with my collaborator and I are at this moment writing a paper about this specific question of value trade-offs in genomics, is that a lot of the discussion and the recommendations that I've been seeing on this field are to focus on precision. So let's say if I'm going to represent human genetic diversity, I have to do it precisely. That means I have to represent it in a continuous way instead of as in discrete clusters, because it's more precise to represent variation as continuous. Now, I think it's interesting to focus on precision in the sense that, yes, it's more precise if we talk about similarity or relatedness than genetic ancestry groups. But at the same time, precision, which is an epistemic value here, often comes at the cost of other epistemic values that are equally important for sciences, such as generalization and simplicity. So as I said, in the recommendations of the National Academies of Science Committee, there was a huge focus on precision. And in fact, after attending those meetings and thinking about it, I was just like, well, but, what would it take to trade things like the ability to generalize and simplify results to focus on precision? Like, is this a decision that has to be coming from the individual level, or should there be like external incentives to do so? Because I can't really think of reasons why an individual would want to trade off generalization and simplicity over a precision, given that it has implications for the sharing of results, sharing within the scientific community, use and reuse of databases, and very importantly, an aspect that we can't neglect, the fact that if you start using new labels in a paper, you won't be cited as much as, you know, using existing labels and existing systems. So I think this is a practical question that needs to be addressed and thought of. Another question is to think about what does it mean to do socially responsible genomics, right? Because we know that there's a very concerning history of race science in genetics and so on. But what I've seen is, I think we're in a really important step of an increasing recognition of slippage between the statistical labels and the social labels, and calls to shift from genetic ancestry groups to similarity and relatedness. This is the main claim in Koop's 2023 paper, and I think this came out many times in the committee about the use of population descriptors. The views were not unanimous. I mean, some were really saying we should keep using ancestry, but I saw a lot of panelists saying, well, you know, we know that ancestry is not the best descriptor, and, you know, perhaps similarity and relatedness would be better terms in this case. So I think that this shift is already, we can already see it, and I'm curious to know, you know, what the practical consequences will be of that. And the final point came with discussions with colleagues where we were thinking, well, you know, many of the recent recommendations, they focus on scientists as individuals, when in fact, for more precise results and better science communication of those results, it's unrealistic to place all the burden on individual scientists. We need to think about it at the political and more institutional level. So what kind of constraints are funding agencies imposing? What kind of constraints what are census requirements? So what is, for example, in the US, there are some requirements to which categories you're going to use, or recommendations of which categories to use to represent diversity. How are we thinking about representing diversity? It's not just a matter of scientists worrying about precision. It's also a matter of what is the ecosystem in place so that these things can happen in an efficient way. And I think that sometimes a lot of the burden is placed on individual scientists, but we should be thinking about really the more macro research ecosystem, where we know that research depends on funding, agencies, on governments, and on policy recommendations. So this is quite abstract at the moment, but I'm still kind of working on those reflections. So for conclusion, I hope to have shown that philosophy of measurement provides a really critical lens to examine this relationship between theory and measurement, as I talked about in the problem of coordination. Second, I hope it's clear that, you know, measuring ancestry is really not a straightforward empirical task. It's deeply enmeshed in other theoretical understandings and concepts like population and genetic similarity. And addressing these challenges can rely on successive stages of iteration, which are already seen taking place. And I'm interested in those stages of iteration to kind of see what is the nature of this iteration. Is it, for example, enrichment? Or are we talking about self-correction? But addressing these challenges requires solutions at different scales, with a special focus on institutional incentives and policy implications. So I think those are the main kind of takeaways from the research that I've done on this so far. There are a couple of acknowledgements that I'd like to make. First of all, I want to thank my two collaborators, Abigail Nieves Delgado and Celso Neto. Abigail is at Utrecht and Celso is at Exeter. They're both amazing philosophers of science, and we have a reading group together, and most of my ideas came from these discussions with them and their feedback. So I would like to thank them. I would also like to thank François Papal and members of the reading group in History and Philosophy of Biology in Quebec. It's a reading group that we have for the Francophone research community here. There are many philosophers of biology in Quebec, and it just started, and it was François' initiative. So really great to have to exchange with my colleagues who are in the same province as I am. And I would like to thank the Fonds de Recherche du Québec Société Culture, which is the funding agency for my very generous postdoc. And I'm really happy to be starting this project. And also thanks to Alan again for organizing, and to Maria for agreeing to comment on my work. And now I will just end my screen sharing. Thank you very much.
Thank you, Yasmin. So I am happy to introduce Maria Nieves Colon, who is in the Department of Anthropology here at the University of Minnesota. Her laboratory uses ancient DNA and modern genomics to study population history, specifically tracing the genetic histories of the Caribbean and Latin America. Maria.
Okay, thank you for that introduction. Hopefully everybody can hear me, and if not, please.
Yes, you're coming through fine. Yep.
Okay, great, wonderful. So thank you, Yasmin, for that intervention. I thought it was really interesting, and there were some echoes in what you mentioned with some of the work that I do as an anthropologist who uses genetics and measurements in genetics to make inferences about human evolution and human history, but also because I work and I interface with human communities, and especially communities that have been historically underrepresented in science, and may come to research with their own ideas about what labels we should use and how we should define and categorize. Some of the issues that you mentioned really speak to the conflicts that we see with that in our own research. So I made a few notes, and I kind of want to touch upon some points that you mentioned that really stood out to me. So one point you mentioned today in your talk, and also in the draft writings that you sent me, was the sort of relative nature of defining genetic ancestry categories, right? When we think about genetic similarity, we inherently have to compare genetic diversity in one group to another group. And that is what gives us this categorization framework. And that made me think about how when we do these types of studies, we often have sort of different levels. So we might be looking at populations, comparing genetic diversity at a continental level, meaning we take everybody within a continent and make them one group. But then later on, we might do some iterative analyses looking within a continental group, like a subcontinental category. And so just to give you a concrete example, we work in my lab with Afro-descendant populations. And so oftentimes when we're trying to estimate these genetic similarity categories, we first want to know, okay, where in the world do these communities share some genetic diversity with? And so we do this sort of continental approach, and then we dive into that continent to say, okay, where within Africa, where within Europe, or where within the Americas? And so it's really this analysis that can become very wide or very targeted, depending on what the researchers are trying to find out, or the communities may want to know, depending on how close you're working with communities. And so that was something that I thought called a lot to how we actually do these studies. Right? We start big, we go small, then maybe expand it a little bit more. And that also brings me to a point you made about the lack of uniformity across studies about what constitutes a population. You know, defining a population is such an interesting problem, and there's so many inconsistencies depending on the goals of the study or what is relevant in that moment. And to give you an example from my own work, so I work in Puerto Rico, which is where I'm from. Puerto Ricans are heavily sampled in human genetics research because of all the reasons you mentioned, the sampling bias, right? The ease of sampling some populations versus others. Some populations are more interesting because of their genetic history for certain types of studies. But most of those samples sort of group all Puerto Ricans together and don't necessarily take into account some local variation that exists within the island. And so right now we are working with Afro-descendant peoples within the island of Puerto Rico who feel like their experience, their history, their evolutionary path has kind of been ignored in these broader samples. And so for that specific study, we have these two samples of Puerto Ricans who just identify as Puerto Rican, and then Puerto Ricans who identify as Afro-descendant, even though maybe an external researcher wouldn't make that distinction. And so those sort of categorizations would be lost in other types of studies, and may not be relevant for other types of studies either. So just kind of an illustration of how that is a dynamic process. I also wanted to comment on a point that you made about the lack of representation in who is being sampled, but also who is doing the science. I think there's been a lot of conversation, as you mentioned, about the lack of diversity in genomics and what is that going to mean for the future when we have precision medicine approaches. But I think also as we move into a field where we're actively trying to recruit participants from underrepresented communities, the lack of researchers from those communities is also going to become a problem. It is a problem because it risks sort of perpetuating these extractive cycles that we know have existed in our field. And that is, I feel, a huge challenge as we move forward, right? We need to increase participation of people that haven't been included, but we also need to bring those people on to be the ones that are designing these studies and maybe using more insider perspectives in defining populations and defining how peoples are going to be, to a certain extent, compared. I really, really liked your idea of enrichment and self-correction for the field. I like that it's an idea that takes into account the history of how these categories come to be. You know, the whole idea of using discrete labels to represent variation is such a problem in biological anthropology for all kinds of phenotypic genetic measurements, but it's also something that we contend with when we're trying to describe the natural world in general, right? What is a species? Where's the line between species? And so thinking a little bit more broadly about how can we enrich the way we're thinking about our field? How can we go back on these categories and iteratively modify them? I think that is a wonderful approach to think about, you know, not sort of thinking out the baby with the bathwater, as you would say. Finally, I do have one point, well, two more points. One is that when we think about clustering methods, I really liked the sort of critical perspective you provided to that. We do have other approaches that we also use to measure genetic similarities, some of which are, you know, precise statistical tests or other forms of modeling. And one thing that I always tell my students, and that I think other geneticists also say, is that, you know, clustering approaches should always be a first exploratory pass. You shouldn't base all your assumptions or your inferences on that because they are by nature exploratory and by nature prone to storytelling, like you said, right? And so that is great for us as geneticists to think about, but unfortunately clustering approaches and those very colorful representations that we make are the ones that make it into the papers, you know, in the cover of the press release on your paper. They're the ones that the public sees when someone is talking about your paper. And so, you know, it's you know, some people might say, oh, well, that's not the only test I do. Well, that's true. But then when we explain it out in the world, this is mostly what people see. So we do have to be careful about the message because DNA does have sort of this outsized influence in the world we live in. People think about DNA as this final arbiter of truth. And so I think that really speaks to how careful we must be about being explicit about the labels that we're using and being cautious of assigning any sort of implicit hierarchy or value that is laden with ordering to those labels. So those are my thoughts. I want to thank you again for your wonderful presentation and for giving me the chance to read through your materials and talk about your work today. And hopefully there will be some cool questions.
Thank you, Maria. So now we're going to move into a time of open question and discussion. We're going to give Yasmin the first opportunity to respond to anything that she wants to from Maria's commentary. And then after that, you can ask questions either by raising your hand using the raise hand function, or you can put a question in the Q&A function of Zoom, or you can send it to us as a chat message and I will read it out loud. Any of those is fine, whatever you prefer. But Yasmin, you have the first opportunity here to respond to Maria's comments.
Hi, so Maria, thank you so much. I made loads of notes on your comments. So I'm really interested in knowing more later. Maybe we can talk later, you know, about your how you kind of implement this interface with actual human communities. And, I am completely aware of this perpetuation of, extractivist science with DNA and all of those, this idea that some communities are kind of, very precious because they have this kind of a type of DNA that's interesting. And I think that also perpetuates a very kind of colonialist, a kind of a colonialist approach to science, which you put very well. It's not just a matter of the representation of the people we're sampling, but also the representation of who is doing it, for what interest, for what purpose, and even for what commercial purpose. So I'm really interested in that more kind of social and political discussion. And in general, well, I'm glad you like the idea of enrichment and self-correction, mostly because sometimes these concepts are kind of it's philosophers of science being a bit abstract about the ideas that we're presenting, but when it resonates in the field, that is really cool, because I would love to talk more about how would this happen in practice, right? What does enrichment and self-correction actually mean? And my final point, which is just yes, I mean, of course, I didn't have time to focus on so many different clustering approaches, and I'm just starting to get into those. But I would be really curious to know later, maybe if there are any examples of where actually in the studies the clustering is just exploratory, and because often what I see is the clustering is, as you said, it's what gets out there. So I'd be curious to know if there are specific examples of this, because I think that would be really, really fruitful to incorporate in this kind of work that I've been doing. But finally, just to say, I really liked the way you put the study when you work with Puerto Ricans, like how do they, whether they self-identify as Puerto Ricans or Afro-descendants. We'd be very curious to see also what are the results of using this kind of population, let's say, separation or distinction, because of course there's not just the scientific part, but there's also a very relevant part about how do people constitute their own identities through genomics. And it seems that you are concerned with those things too, and I'd be really, really interested to chat more. But thank you so much again for commenting on my work, and really the work your lab does really looks amazing.
Okay, so let's take a first question from the Q&A function from Kino Zhao. And here it is. I'm curious, in what way do you think methods such as clustering count as measurement, as opposed to, say, data analysis or modeling? Insofar as the goal of measurement is representation, then the fact that genetic ancestry is continuous seems to suggest that the target of categorical measurement just doesn't exist, and so the effort is wrong-headed. I'd like to hear more about the motivation of conceptualizing this as a measurement issue.
Thanks. Yeah, so I've dwelled on this as well. And I think that the question here is that ancestry is a proxy for measures of similarity. So maybe really what I'm talking about is how do we measure similarity? But at the same time, but the question of using proxies to me is how I came into measurements, because in the end we are using proxies to represent some aspect of reality. Well, let's not go into the whole realism debate, but in this specific case, I feel like it's relevant to talk about clustering as methods to represent and measure genetic similarity. But I'm still not 100% convinced that this shouldn't just be measuring similarity. I'm still kind of thinking about it, and I guess I'm using measurement here in a kind of a broader, let's say, a broader definition of measurements that is just interested in how am I going to represent an aspect of reality while interacting with this aspect. So I think that would be my initial answer.
Okay, so let's move on to a second question from Jerry Chandler. Jerry, you should be able to unmute yourself and ask your question.
Okay, yes, thank you. Enjoyed both presentations, very interesting. I would like to raise, I think, what's probably a deeper philosophical question about the general approaches being used. And so the issue is one that I think expands all the ranges of science. And this is, of course, the question of the distinction between continuity and discreteness. And in this context of genetics, although it's, they say it's a essentially universal question across all of the sciences and all of the languages of science, and that is by nature, a single nucleotide polymorphism is a discrete thing. It must be discrete, and the measurement technologies and methodologies that are used to measure it are reproducible, they're consistent, they're sound science, if you would. And so the sudden shift to then use of continuity in the notion of how you're going to analyze populations creates this deep conflict of meaning. And so it's more of a comment than a question, but if you have, I'd be interested from a philosophical perspective, how you view this challenge that penetrates all of the sciences and not just your particular problem. Thank you.
Yeah, so thank you so much for that question. And I've thought about this as well. And I think that, I mean, I don't have a specific answer of, you know, when this continuity and discretion starts. But the way that I tend to look at it is that depending, specifically in genomics, there are pragmatic reasons to go more or less fine-grained in the categories that one is using. So in this case, you can see it in different kinds of studies. So some studies will have, you know, just very broad continental regions, like Maria was saying, but others will go more and more fine-grained. And ultimately, I think the idea is the more fine-grained we go in the divisions, the closer we are to representing continuity. But I think that when we are measuring the differences, let's say, between two individuals, then we are talking about something that in the order of discrete, not continuous. So I think, yeah, that would be the way that I would look at it in terms of grain. So are we talking more fine grained or less fine grained? And ultimately, we will reach a point where, you know, the ultimate level of analysis will indicate a discrete difference. Yeah.
Okay, so thank you, Jerry. Let's move on to next question from Corey to chair. Corey, you should be able to unmute and ask your question.
Wonderful. Thank you very much for the talk. I had a question about, well, about why you weren't saying to get rid of these ancestry groups, because you were very clear that you didn't want to say that we should get rid of them. And the reasons that you gave maybe about halfway through the talk sounded like largely pragmatic reasons. Like there are really good pragmatic reasons to keep these around because they help us organize data and you know, communicate across different scientific investigations. The very good pragmatic reasons. But it seems to me like, you know, it just on general principles, if there are real epistemic problems with these groups, then those pragmatic reasons are likely to, you know, are more likely to mislead us than they are to actually help. Like we'll organize data and that'll be really helpful in some sense, but we'll be organizing data around the wrong things. And so like while there's some sort of pragmatic gain there, it's actually sort of more epistemically costly than beneficial. So I was wondering if I've just misunderstood like what kinds of pragmatic gains you're thinking about, or if you think that there's some reason to think that we can have these pragmatic gains even if there's some real epistemic problems with the ancestry concept.
Yes, thank you so much for this question, Corey. I've been really thinking about this, and I think it's a matter of how much I think the pragmatic reasons are significant for scientists working in the field, and I think they are really significant to the point of being okay with some epistemic losses. There are a couple of there's a really interesting paper by Rena Bliss, and I forgot the exact year of the paper, but it's Bliss and Panofsky, I think 2017, where they explain why, why there's so much vagueness in the use of things like ancestry categories, and it's an ambiguous term, and sometimes this ambiguity is on purpose. And one of the reasons that they mention is that, like I said, there's just little incentive to rethink those clustering, how we cluster, and that the main reason for keep using them are the pragmatic reasons. And that opened a light in my brain, because in some ways in philosophy of science we tend to think, well, the reason to talk about those clusters is probably because they exist out there, or that there's something, like, let's say, in nature that is that we can represent through these clusters. And in fact, in scientific practice, that's not the question that's being asked. The question that's being asked is, how often will my paper get cited if I keep using this, if I keep referencing this? How will this increase the visibility of my work? So I decided to put those pragmatic reasons more front and center, and move a little bit away from the more ontological questions. Now, I'm aware, as you said, that it is more epistemically costful, but I do think that the pragmatic reasons are strong enough that they prevent us from getting rid of those labels. And I mean, as a more kind of practice-focused philosopher of science, that's the part that I would like to explore more. But for sure, there are ontological contributions to this discussion as well. Yeah.
Okay, thank you, Corey. So further questions? Again, you can use the raise hand function, or you can drop them in the chat, or offer them in the Q&A function. Well, while we're waiting for other questions to bubble up, I have one that is kind of connected to what Corey just brought up, which is, it seems like one of the things that is maybe the most challenging to think about for the use of the categories for organizing data is that it has, in some ways, permanently shaped some of the legacy data that exists. And so then the question in terms of epistemic costliness might be poignantly put as, when do we just need to get rid of legacy data instead of trying to find ways to leverage some kind of back compatibility that would give us some use of it? Maybe there are instances where we just need to say, no, we shouldn't be using it. And do you think that some of the considerations that you're bringing might give us insight into that? That is, that over time, as you are improving these methods, is part of that procedure include removing some of that material that was used in the past?
That's a great question. And I think removing is just maybe not realistic. It might be a kind of, in a way, a naive solution that would never really be pragmatically put in practice. But I think that I think that deliberation here is important. And one of the things that the National Academies of Science Committee was doing was precisely that, right? Was to talk about how those labels are being used, whether they should continue to be used. Now, to me, the critical point is not whether we should scrap that and start over, but in those deliberations, how are they going to be put into practice? So what is actually going to change? What is actually going to be self-corrected? What is actually going to be enriched from those deliberations? Because sometimes I find that there's a gap between the theoretical deliberations and the more practical implementation of those. And the idea here would be I don't think there's ever a point where we should just need to get rid of the legacy data, but I think there's a point where we should go over it and at least be transparent about why this is being used and why those labels are being perpetuated. And transparency was one of the big things in this report that came out, which is in the papers or in the way that the science is being published, to explain the rationale for the decisions. Why am I using this as a reference? Why am I using these ethnic or ancestry categories? And I mean, it will take time, of course, but I think that's maybe a good way to think about it, instead of just getting rid of the legacy data. But I don't know. I'm open to disagreements on that.
Yeah, no, you might think of that as the nuclear option, not one that's going to be often taken, but it did seem like it was at least a possibility on the account that you offered. So good. Other questions? Anything else for Yasmin? Maria, go ahead.
Hopefully it's okay that I also raise my hand.
Yeah, no, please jump in.
I just wanted to kind of comment / question that some of these categories of ancestry that we use to explain the world we live in today are now also like finding their way into ancient DNA studies. So sometimes we use these categories like, let's say, for example, French or English, which are like modern nation states, but then they also kind of are used in these studies to explain ancient migration patterns in ways that make sense to people today. And so my question for you, Yasmin, is how do you think this sort of dissonance between how these labels are made also impact the way we think about the past, as sort of like our path as a species?
Yeah, I think that's a really good question. I don't know that much about ancient DNA research, but I'm kind of getting into it because it's really fascinating. But it does make sense what you said, that some of the categories that are used like now to make sense for us now, that those are the ones we have to use, like, for example, French and English. So honestly, I don't know that much about ancient DNA research, but I would be really curious to know how those categories are deployed. In her book, Catherine Nash, she makes a similar she talks about a similar notion, which is, these geographic labels that we use, they look neutral because they are, for example, continents. But in fact, one of her points is that any geographic label is politically situated, is socially situated, and that the way that we use them will, like you said, perpetuate some structures that may not be the ones that we want to. I would have to think more about that specific aspect of ancient DNA research.
Okay, Lauren Wilson is up next. Lauren, you should be able to unmute and ask your question.
Okay, can you hear me? All
Yeah.
right. Wonderful. So your answer to the last question actually kind of anticipates my question, which is when we're looking at either the development or the continued use of ancestry categories based on geopolitical boundaries, to what extent should certain other values influence their use? Say, kind of standard geopolitical conflicts, which nation states are often doing this type of research, which kind of political agendas might be wrapped up in certain scientific investigations. I'm wondering if you've, looked into that and what your thoughts are.
So I haven't looked specifically into that, but one thing that I've looked into is a controversy about what's called the Human Genome Diversity Project, and there's an excellent, quite a long paper by Jenny Reardon, where she explained, the way that the geneticists in that study that were aiming to collect DNA from certain indigenous populations kind of related to those populations and how politically problematic that was, and in the end, that there was so much controversy with that study, because, like Maria was saying, there is a kind of bias towards who is conducting the studies, what worldview they have, what political worldview they are in, and how these are going to structure inquiry in forms that are going to perpetuate some extractivist science, so like just go to a place and collect this data, and how this group is going to represent another group that wouldn't necessarily represent themselves in the same way. So I think this is quite an important issue that I've come across, but still kind of trying to learn more and keep up with. But I think those questions are really important.
Okay, let's move to next question from Dana Mathiasen. Dana, you should be able to unmute and talk.
Okay, thanks. And thanks for the talk. I kind of want to go back to one of the first questions that was raised in the Q&A box and just ask whether you think it might be useful in a case like this to try and distinguish what we might call sort of proximal and distal measurement, or sort of think about stages of measurement in terms of the kind of theoretical or inferential load that they take on, because it seems in a case like this we can distinguish several stages. One in which you sort of use basic genome sequencing, gathering samples from individuals. A second one in which you are developing some method for comparing those sequences. A third in which you are then sort of clustering based on a kind of higher order comparison. And in each case, there's sort of, it seems to me, more theory being loaded in. And so, yeah, I wonder what you think about, I guess, sort of decomposing this idea of measurement into these stages, and whether that might be a kind of analytically fruitful approach.
Yeah, thanks for this. I think it will be really useful. And in another paper that is currently under review, I've tried to do a little bit of that with kind of like a workflow of the different stages where the assumptions come in. And I think it's a really useful strategy. It is sometimes confusing because there are so many different techniques. And like Maria was saying, depending on the type of study and the goal of the study, these techniques will vary. But I'm definitely interested in how that happens in admixture studies and genome-wide association studies. And it is always useful to break down. And clustering is just a part of it, right? Because after there are going to be other steps before that and after that, especially in the interpretation of the data and the analysis and what's going, like how it's going to be used. So that's one thing. And another field that's quite interesting where philosophers are starting to take interest in those is the relationship between how polygenic risk scores are measured and actual outcomes are measured. So I think this is like it's like a parallel problem, because a lot of these many of these studies are interested in establishing polygenic risk scores. So yes, I think that's a useful strategy to try and distinguish the different stages.
Okay, final questions that folks have. All right, I think we may have reached a good point of closure. So let me first and foremost thank Yasmin for a wonderful presentation and for your response to questions and engagement with these issues. It was really fun to see it, and I learned a lot. Maria, thank you so much for taking the time to be a commentator. It was really rich to have your perspective, and I think added a lot. And thank you to everybody who was here with us and joined us today, asking questions and listening in. We look forward to seeing you again on another occasion. Our next installation of the International Postdoc Forum on the Philosophy of Science will be in just under 2 months' time at the beginning of April. All the details are on the Minnesota Center for Philosophy of Science webpage. Check it out there. Otherwise, everybody have a good rest of your day. Take care. Bye-bye.